In these sections, we will review alleles, single nucleotide polymorphisms or SNPs, genotypes, and haplotypes. Let's take a look at this example. Here we have a child who has inherited chromosome 7, one from mom and one from dad. The DNA from mom has a sense strand here and an anti-sense strand, and the same is for dad. The sense strand, you can think of it as the strand that makes sense, is the one that carries the code. So we look at position 5, counting from left to right, 1, 2, 3, 4, 5. Here we have, in position 5, this allele C. And let's say that this allele C is the least common, and this is more of a variation in the DNA. So this one is a SNP right here. In position 5, in the chromosome from dad, the child inherits the T allele. So this means that the child is heterozygous at locus 5, and his genotype is CT, C from mom and T from dad. Here is a quiz. Here we have 6 individuals and a particular locus on chromosome 2. Each individual has 2 pairs of chromosome 2, and these are the alleles that that each individual carries. Which one or ones, if more than one individuals, is homozygous for the highlighted SNP? You can hit the pause button and think about it. So here is the answer. Individual 2 is homozygous, and individual 3 is homozygous. The other individuals are heterozygous. Let's look at the SNPs in more detail. Coding SNPs, abbreviated as cisNP, are located inside the exons. Exons are the parts of DNA that code for proteins. In between exons, you find introns. These do not code for a protein. So the strand of DNA is transcribed into RNA, which then undergoes additional modifications like splicing, and then you have now a final mature mRNA which contains only the exons. This mRNA is translated into a protein. So here is the question. Is a coding SNP always damaging? Hit the pause button and think about it. The second question, is a non-coding SNP always benign? Hit the pause button and think about it. So here's the answers. The answer to question 1, is a cisNP always damaging? Well, that depends. The cisNP will be in the exon, and then it is transcribed and translated into protein. However, what if the cisNP is happening in a position where eventually becomes an amino acid really deep inside the protein? In this case, it might have little effect on the function of the protein. In addition, the cisNP could maybe there could be a change in the codon, but the amino acid might actually remain the same. So the cisNP could be synonymous, where you have, yes, you have a change in the DNA, but at the end, the amino acid will remain exactly the same. And in that case, you do not see any difference. For question number 2, is a non-coded SNP always benign? The non-coded SNP occurs in the introns or in a promoter region, so any area where any loci that are not part of the exons. So you would think that they might not have an effect. Now the introns, if a SNP happens in an intron or in a promoter region, it could actually damage the signals that help produce the protein, and also there are now, we know now that the introns are not just junk DNA as we used to think of, but they might contain important regulatory regions. So a non-coding SNP is not always benign. Let's see haplotypes now. Haplotypes are a set of closely linked genetic markers present on one chromosome, which tend to be inherited together. So here's an example. We have the sense strand of 6 individuals at a particular chromosome. So these are the same loci for each individual, and you can see that for the most part these individuals have a similar genetic code. However, you can see, for example, on this first row that this individual has a allele at locus 4, a G allele at locus 12, and a T allele at locus 15. As we look at the other individuals, they have a different set of alleles. We can think of this set of 3 alleles as an haplotype. So the first individuals as an AGT haplotype, the second individual has a GTA haplotype, the third has an AGT haplotype again, and so on. So at the end, among this group of individuals, there are 3 possible haplotypes: AGT, GTA, or AGA. Here is a question. Why are haplotypes of interest in genetic epidemiology? Hit the pause button and think about it. The answer is that this may actually be time-saving and cost-saving opportunity. If we know that, for example, an individual at locus 4 always has, if an individual at locus 4 has an allele A, and because it's closely linked, then we might guess that at position 12, this individual would have a G allele. As you can see in this case, individuals who have an A allele and locus 4 will have a G allele at locus 12. So these are tightly linked. And so this means that when we do a genetic analysis, we might only need to look at one of the 2 alleles, maybe A, and then we can guess the second allele, G. So we do not have to run the entire sequence of the DNA, but we can just select specific SNPs, which we called tag SNPs or tagging SNPs, that will actually give us information about entire haplotypes.